Okay, welcome back to Affinity Digital again. Myself, Usha, your botany teacher. I hope you have done your questions well, and it's giving you much benefit in this short period of 2 months before your exam. So anyways, let's start with a discussion. This is test series number 4. So the first question is, alternate phylotext C is present in all except. This should be in the case of guava. Guava will have opposite. It will have opposite superimposed taxonomy. Phylotexy, sorry. So opposite superimposed phylotexy. All the others will have alternate. So answer is 4. Next, the development of the embryo is towards, this should be micropylar N. Of course, if this will be ovule, in any case towards the micropyle site only the egg cell will be present. So since the egg cell will be present and then synergid here, pollen tube will come towards it and the 2 male gamete, obviously it will fertilize with this. That is why zygote or the further development leads to embryo, that will be always towards the micropylar N. Next, from the given examples, penicillium, these are the example. How many produces sexual spores exogenously? How many produces sexual spores exogenously? So in this, let's see this agaricus, astylago and puffball. In this 3 case, these are basidiomycetes. Okay. So if it is basidiomycetes, then it will have basidium, and in above the basidium there will be presence of 4 basidiospores. This basidium is actually hypha only. The terminal end of the hypha inside the basidiocarp, that means fruiting body inside the basidiocarp, that is called as a basidium. So it is hypha only. So inside this basidium only, it will have karyogamy and meiosis, and after that karyogamy and meiosis only the basidiospores are produced. So when basidiospores are produced, it goes outside this basidium. So basidium is hypha only. So since this basidiospore is outside the basidium hypha, basidiospore is an example of exogenous. So this is an example of exogenous spore. Okay. So answer to this will be 3. But if you say with respect to other fungus, answer is 3 because these 3 are basidiomycetes. Penicillium neurospora. Okay. Penicillium and neurospora will come under ascomycetes. So in the case of ascomycetes, there is the presence of ascospores which are inside the ascus. 1, 2, 3, 4, 5, 6, 7, 8. This is called as the ascospores, which are sexual meiospore, and the region where ascospores are produced is called as the ascus. Again, if you ask what actually is ascus, ascus is a sac, SAC, sac-like structure, and it is a hypha only. So this inside the ascus, which is a sac-like structures, which is hypha, inside this the ascospores are produced. That is why ascospores is an example of endogenous spore because it is inside the hypha. So this penicillium and neurospora will be ascomycetes. But in the case of alternaria and trichoderma, this too is deuteromycetes, and sexual reproduction is absent in deuteromycetes. Inulin is a polymer of fructose. Next is 105. One of the main criteria for 5 kingdom classification is phylogenetic relationship. Next, which one of the following have both chlorophyll A and C? This should be laminaria. in the case of algae, green algae will have A and B, chlorophyll A and B. Okay, so remember like this, chlorophyll A and B, green algae. And if it is red, then red will have chlorophyll A and chlorophyll D, will be in the case of red. And if it is brown, then it will have chlorophyll A and C. So chlorophyll A and C is a character of brown algae. So when we say Porphyra and Gracilaria, this will come under red algae. Porphyra and Gracilaria is red algae. Red algae should have chlorophyll A and D. ATS type of ribosomes are found in cytoplasm of eukaryotic cell, while chloroplast, mitochondria, prokaryotic will have 70S. The seed of lupine germinated and flowered after an estimated record of 10,000 years of dormancy. This is correct. Okay. This is taken from Sexual Reproduction of Flowering Plants, class 12 part. These are all direct NCERT basis. 2,000-year-old viable seed is of Phoenix dactylifera. This is also correct. Phoenix dactylifera, the common name is death palm. So the seed is viable for 2,000 years. Means viable means ability to germinate. Oh, that is for 2,000 years. This is correct. Dehydration and dormancy of mature seeds are crucial for storage of seed. This is also correct. So dormancy is required and the dehydration is required. If the seed is dehydrated, means moisture contains less, then only you can store it for longer duration. If it has moisture, of course it will start germinating. That way it will get spoiled. So for storage of seed, dehydration and dormancy is required. So what about the next one? The seed formation is independent of water. Obviously seed formation will depend on water. When there is a, not directly water, but in the process of fertilization, when plant has to grow, flower has to produce, fruit, seed, how it will form, obviously it will require water. So this sentence should be, this sentence is actually wrong. It should be, it is dependent on water. That means 1, 2 and 3. Xylem parenchyma has stored food materials and tannin. This question, even though it is deleted, I will suggest you to study at least this part of simple parenchyma, simple permanent tissue parenchyma, collenchyma, sclerenchyma and xylem and phloem. The basic things, it still can be asked. So this question has put up here like xylem parenchyma. This is still, it is applicable. Even though it is deleted, the basic concept still requires. 110, the ratio of the 9:3:1 and dihybrid cross denotes that this should be the alleles of two genes are segregating independently. This is law of independent assortment. What does law of independent assortment says? When the two pair of when the two pair of traits are present in a hybrid, this is one pair of trait, this is another pair of trait. Two pair of traits are present in a hybrid. The segregation of a pair of character. This is one pair of trait, this is another pair of trait. That means this will be round and wrinkled and this should be yellow and white. So now this two will be having this and this two allele will be this. So now this is a pair of allele here, this is another pair of allele here. That means the alleles of two genes, these two are the alleles of two genes. This is one allele, this is another allele. So these are two alleles of different gene. And this again same condition, this is one allele, this is another allele of different gene. They will segregate independently. So the line is written like this. The allele of two genes are segregated independently. Allele of two genes. This is one allele, this is another allele. This again is one allele, this is another allele. So the allele of two genes are segregating independently. This is what it says according to law of independent assortment. And the two genes which follows the laws of independent assortment in a dihybrid cross, they will have 9:3:3:1. Okay, so whenever we perform dihybrid cross, and if the dihybrid cross comes out to be 9:3:3:1, that gene is unlinked gene, means they are totally not connected with one another, and it will always have the ratio phenotypic 9:3:1 if it is unlinked. But when there is a linkage, like here in this first option, there is a complete linkage between two genes. When the gene are linked, then it will never have recombinant character. Complete linkage will never have in recombinant characters when we'll perform test cross. Okay, when we'll perform test cross or when we'll perform selfing, there will be never be recombinant characters. If it is test cross or if it is self hybridization, when we, in the case of the complete linked genes, there will be no formation of recombinant characters. This complete linkage, that means there will be no formation of recombinant characters. So this is only possible when the alleles of two genes are segregating independently. So that means alleles of two genes, like this allele and this allele, they are different alleles of two different genes. They are segregating it independently. Okay. And if it is a case of multiple allelism, of course, it will not have this ratio. Plant families like Convolvulaceae and Solanaceae are included under polymonials. This is a family which includes an order polymonials. Okay, this one, the color is given here. Like a carotenoid will have yellow to yellow orange. That means A will be 3. A will be 3. And then B will be 1. Xanthophylls will be yellow color. B will be 1. Okay. And the C will be 4. Chlorophyll B will be yellow green. C will be 4. C will be 4 and D will be 2. 2 means bright or blue green. Chlorophyll A will have bright or blue green. D will be 2. D will be 2. So A will be 3. Okay. B will be 1. C will be 4. D will be 2. Option number 3. Next, tailing. Addition of the, you have to find out incorrect matching pair. Addition of the adenylate residues at 3N. This is correct. In the case of eukaryotes, at the end of the hnRNA, at the end of hnRNA, first it will have methylation or capping. Okay, in which a methylated guanosine will be added here at the 5' site. So this will be methylation and capping, and at the 3N site there will be the presence of the 3N site, there will be presence of adenine residues, polyadenylation. Adenine will be added, about 200 adenine residues is added at the 3N site by enzyme poly-A transferase enzyme in a template independent manner, means without using a template. So this presence of polyadenylation, this should be at the 3N site. This is correct. At the 3N site. This is in the case of eukaryote, and it will be added at the hnRNA. First at the methylation or capping at the 5' end and the polyadenylation at the 3' end. Represents the dominance of the RNA wall. Splicing means in this, what happened from the hnRNA, the impure form of the RNA, from it the introns are removed. That removal of the introns and presence of the ribonucleotides in the mRNA, that is a phenomenon which shows the dominance of the RNA wall. Splicing is also correct. Matured RNA is a process snRNA. Till methylation and capping and polyadenylation, Till here, the RNA will be called as an hnRNA, or heterogeneous nuclear RNA, because it consists of both introns and exons. Just after the process of splicing, after removal of the introns, then it will be called as the mRNA, or process messenger RNA. Okay, so mature RNA means after hnRNA is properly processed. So capping, unusual nucleotide added to the 5N of mRNA. Unusual nucleotide added to 5N of mRNA. This should not be mRNA, it should be hnRNA. And it is, saying unusual nucleotide means it is a guanine. It is a guanine only, but in the guanine, at the 7th position, it consists of a methyl group. So that's why you can also say it to be a form of methylated guanosine. Okay, so it should not be mRNA, it should be hnRNA. That's why the term is used, unusual nucleotide. Means it is a guanine which has a methyl group, so methylated guanosine. That is why it is called as an unusual nucleotide. This term is correct, but it is not mRNA, it should be hnRNA. So answer will be this 4. 4 is answer, capping. Once the biochemical oxygen demand BOD in sewage is significantly reduced, the resulting flocks are allowed to settle, and this sediment is known as activated sludge. Okay, and this activated sludge will be sent to a biogas plant. And when we say primary sludge, this will be the initial, like the plastic bags or plastic bottles, the solid particles which is first formed in the first process of filtration process. That will be called as a primary sludge. So answer to this will be activated sludge. Okay, here it is given, we have to find out how many are incorrectly matched. Incorrectly matched. RBC is biconcave and oval. It should not be oval, it should be round. These are all strictly NCERT. You will find this in the NCERT pages in a cell part. Biconcave and it should be oval. It is given here oval, it should be round. And the mesophyll cell, it is not elongated. Mesophyll cell can be either round or round and oval. Okay, so that means this is incorrect, this is also incorrect. Nerve cells should be long and branched. Nerve cells should be long and branched. So, but white blood cells amoeboid, this is correct, correctly given. So 1 is incorrect, 2 is incorrect, 4 is also incorrect. So 1, 2 and 4, question is asked incorrect here. 1, 2 and 4 are incorrect. White blood cells amoeboid is correct. Okay, so this is 3. Free-living microbe, 116. Free-living microbe, this will be 1. A is 1, free-living microbe, azotobacter, and it will fix nitrogen. Non-symbiotically, it is a free-living nitrogen fixer. Glomus will form mycorrhizal association. B is 2, and it belongs to class Phycomycetes, glomus. And the symbiotic microbes will be C3. So A1, B2, C3. A1, B2, C3. Symbiotic microbe rhizobium will present inside the root nodule of leguminous plant. This is ecology question 117. Let's see. Primary consumer is got. This is correct. We have to find out the correct matched. Primary consumer is got. Of course, primary producer will be plants. Primary consumer will be got. This is correct. Detritus is the raw material for decomposition. This is also correct. For decomposition process, the raw material present is called as detritus. And pyramid of number in grassland ecosystem is upright. This is also correct. 1, 2, 4. 1, 2, and 4. This is correct answer. Now see, pyramid of number in grassland. Let's consider, say, suppose let's take plants. Okay, let's take plants, and then next is in the grassland. In grassland, the first one is plants, and say insects is the primary consumer, and let's say frogs secondary consumer, and then let's say snake. Okay, and again it can be eaten by eagle. Okay, so this is a grassland ecosystem. The primary producer will be plants, primary consumer, secondary consumer, tertiary consumer, like this. So if we consider this way, plants will be obviously high in number. Okay, insects will be lesser, frogs will again be lesser, snake will be less. Of course, if you put eagle, it will be 1 or 2, like this. So this is an example of the grassland ecosystem: plants, insect, frogs, snake, and eagle. This is a grassland ecosystem. Okay, so when we put it in the pyramid, ecological pyramid, it will be upright. That is why pyramid of number in grassland is upright. And saprophyte included in ecological pyramid, this is wrong, because this is one limitation of ecological pyramid. Ecological pyramid does not include saprophyte. So 1, 2, and 4. Henking observed that 50% of the sperm in a few insects receive a specific nuclear structure after spermatogenesis. This is direct NCERT line. This is correct. After spermatogenesis, the sex determination in honey bee is based on a number of sets of chromosome and individual received. Is based on number of sets of chromosome and individual received. This is also correct, because in the case of honey bee, haplodiploidy will determine the sex. Will determine sex in case of honey bee. Haploid, that drones are haploid, and the queen bee and worker bees are diploid. So haploid, diploid, that means haploid will be single set, diploid will be 2 set. So the number of set will determine. This is also 119. The loss of certain species in an ecosystem may not initially affect its proper functioning. Okay, this is correct. If certain species are lost in the beginning, initially, this was in an ecosystem, if one species, like say a species of the bees or a species of insect is lost, then initially in the beginning it might not affect it. Correct. The loss of key species that drive major ecosystem function is a more serious threat to the functioning of the ecosystem than loss of few species. Now this one, the sentence here in the NCERT, this term is given key species. Key species means in an ecosystem sometimes there is a particular species which is controlling the species diversity of 2 or 3 other species. Let's consider in an island. Okay, in an island, there is a plant species growing. Okay, so this plant species, what happens is, in the plants, the fruit which is formed, that fruit is eaten by the people living in that area. All the people living in that area of the island is eating that species of the fruit which is produced by this plant. Okay, and now next, that means the staple food of that area. Again, this plant is pollinated by a type of the bees which is present over there. Okay, and again another way, this a particular type of an insect also used to eat these plants. So that means this is an island, an isolated island, and this particular plant species is a very important plant of that area, because one, the people living in that area is eating the food of that. Second, bees is pollinating that area of that plant species. Again, a particular insect used to eat or infest that plant species. So that means the species diversity of this insect, people, bees is controlled by this plant. Okay, so now this plant is a keystone species. This plant is called as a keystone species of that area. So if I remove this keystone species plant from that area, then what happened? Then insect, people, bees, the species diversity will all be declined. That means this keystone species or key species is the main species of that area and controlling the species diversity of the other three species. If I remove that, the whole ecosystem will break down. This is what it is saying here. The loss of key species means keystone species that drive the major ecosystem function is a more serious threat to the functioning of the ecosystem than loss of few species. Okay, so this is also correct, and that is why this is connected. The loss of the certain species in an ecosystem may not initially affect its proper functioning. This is correct, and this is also correct. And why this is explaining, because when the keystone species is declined, at that time only the serious threat to functioning of the ecosystem occur. But loss of few species in the beginning, or one species in the beginning, that means this one, it will not affect much. But if it is keystone species, then it will affect the whole ecosystem. Meaning is that one species will not affect much, but keystone species, then it will affect the whole ecosystem. That's why this is being explained by them. Okay, so answer to this will be 1. 120. Friedrich mixture, F mixture. This is molecular basis DNA as an acidic substance, A2. Watson and the Crick, B1. Wilkin and Franklin will be X-ray diffraction studies, A4, C4, and the Griffith will be transforming principle. 3, yeah. 3. So A will be 2, okay. B will be 1, A is 2, B is 1, C is 4, A is 2, B is 1, C is 4, D is 3, B is 3, 2, A is 2, B is 1, C is 4, D is 3, is 4, and D is 3. Okay, 2. Next, read the given statement and select the correct option. Now, this is statement based. Centromere split during anaphase stage of mitosis. This is correct. During the mitosis process, when the centromere will split during a time of anaphase. Correct. Chromatid separate during anaphase stage of mitosis. Of course, centromere will be split in anaphase. That is why chromatid will be separated. This is also correct. Chromatid separate during anaphase stage of mitosis. 121 will be both statement 1 and A and B are correct. Each trophic level has a certain mass of living material at a particular time called as standing crop. In each trophic level, at a certain period of time, particular time, the amount of the living material present, that is called as a standing crop. And this is not given in NCERT. Answer to it is this, but standing state will be, this should be amount of inorganic substance present in ecosystem for unit time at a given area. For unit time at a given area. When we say standing state, that will be amount of inorganic substance present in ecosystem for unit time at a given area. And when we say standing crop, that will be living material which is present at the trophic level, each trophic level. Standing state will be per ecosystem, per unit area at a given time. And yes, this will be done. 123. In Hershey and the Martha Chase experiment, the viral infecting agent was found to be, this is DNA. 123 will be DNA. Photosystem 2 is located on the thylakoid membrane. This is correct. PS2 is located in the thylakoid membrane. Photolysis of water and NADP reduction occurs in the stroma. Photolysis of water will occur in lumen of thylakoid. It will occur in lumen of thylakoid. And NADP reduction, this does not occur in stroma. NADP reduction occurs in the thylakoid. It occurs on thylakoid membrane. Occurs in thylakoid membrane towards the stroma site. Towards the stroma site. It will occur in the thylakoid membrane towards the stroma site. But photolysis will occur in the lumen of thylakoid. So 124, A is true, but region is false. Great. In sickle cell anemia, mutation arises due to a single change in a single base pair of DNA. It is given here deletion. Sickle cell anemia is due to substitution. And in the substitution, it is because of transversion. Transversion means purine substitute by pyrimidine. Purine substitute by pyrimidine or vice versa. Means purine is substituted by pyrimidine, or we can say pyrimidine is substituted by purine. That is called as the substitution mutation. And that type of substitute mutation is called as a transversion. In the substitution mutation, there are two types. One is transversion, another is transition. Transversion means purine substituted by pyrimidine, or pyrimidine substituted by purine. That is called as transversion. And the transition means purine substituted by purine, or pyrimidine is substituted by pyrimidine. Okay? So sickle cell anemia is due to substitution. That type of substitution is transversion. Okay, this is respiration chapter. PCA cycle. PCA cycle will occur inside mitochondrial matrix. A will be 3. Glycolysis will occur in cytoplasm. B will be 1. ETS will occur in the inner mitochondrial membrane 2. So A is 3, B is 1, C is 2. A is 3, B is 1, C is 2. Next question. The centrifugation technique that is used to differentiate heavy DNA from normal DNA is cesium chloride density gradient centrifugation, which is used in Michelson and the Stahl experiment to prove semi-conservative replication of DNA. Heterotrophic nutrition can be present in how many groups of protista? This should be 2. What are the 2? Euglenoids and protozoans. Euglenoid and protozoans, they will have heterotrophic nutrition. The technology of biogas production was developed in India mainly due to the efforts of IARI or KVIC. E will be KVIC, IARI, KVIC. 129 should be 3. Okay, inhibition of the succinate dehydrogenase. This is an enzyme question. It has come up here in botany part. Anyways, this you must be knowing this. This is a form of competitive inhibition. Next question. 131, it contains water, sap, excretory product, and other unwanted milk. Okay. It is bound by a single membrane called tonoplast. Once when we say tonoplast, we know it is a vacuole. It contains forms cell sept. It maintains a Tugger pressure. The main Tugger pressure is maintained by vacuole in the case of plant. What is it talking about? When we say tonoplast, means it has to be plant vacuole. 132 next. 132. Okay. 132 next. Regulator. You have to find out incorrect statement. Regulatory sequences do not code for any RNA or protein. This is correct. Monocystronic genes in eukaryote are split because it consists of introns and exons. Intervening sequences do not appear in mature process RNA. Intervening sequences means introns. Okay. Answer to this will be this. Very simple. Exons are non-coding and introns are coding. Exons will be what? Coding. Introns will be non-coding. So answer will be 3. 132, 133. Auxin induces parthenocarpy in tomatoes. This is correct. And the 24D does not affect mature monocot plant. This is also correct. Okay. This is a direct NCERT line. It is correct, but it is not connected. So that's why answer will be 2. For formation of the seedless fruit, generally auxin are applied because auxin induces parthenocarpy, means formation of the fruit without fertilization. Okay, 134, habitat lost. Habitat loss, major habitat loss, it occurs in a tropical rainforest. So A will be 3. Then the steer cow, this is a totally extinct species, B1. And the lantana will be alien species inhibition, 2. So A3, B1, C2. A3, B1, C2. 2. Okay, next. 135. During cytokinesis, the furrow formation does not take place in plant cell because of the presence of inextensible cell wall. Now, to form furrow formation occurs in animal cytokinesis. So, animal cytokinesis means the cell membrane will cleave towards the cytoplasm. The cell membrane will cleave towards the cytoplasm inside. That along, I'm saying I better explain it. Suppose during the time of animal cytokinesis, this is cell membrane. Below the cell membrane, there is a presence of the cytoskeleton. Cytoskeleton will be present. And what is the cytoskeleton present here? Which is called as the microfilament. So this microfilament cytoskeleton along with the cell membrane will help in cleaving towards the microfilament, which is a type of cytoskeleton along with the cell membrane, will cleave towards the cytoplasm and form a cleavage furrow. This structure which is formed here is called as a cleavage furrow. So the cell membrane along with the microfilaments cleave towards the cytoplasm and form a furrow. So this method is possible only when cell wall is absent. Okay, that means in the animal. So in the case of plant, it is not possible because plant has inextensible cell wall. Next, 136, linkage results in a lower frequency of recombinant phenotype. This is correct. When there is linkage, then recombinant phenotype will be less. Linkage is caused by gene being located on different chromosomes. Linkage means physical association of the two genes and they should locate on the same chromosome. Here it is given different chromosome. This is wrong, but this is correct. Linkage results in a lower frequency of recombinant phenotype. If assertion A2 is true, but reason is false, will be the answer. 136 is 3. Incorrect matching pair. This should be adiantum. Adiantum should be homosporous. Adiantum is called as a walking fern because it is vegetatively propagated by leaf tip and it is homosporous. 138. Marine form of the protozoan that have silica cells on the surface are, they should be amoeboid protozoans. 138. Next. Assured seed set, even in the absence of pollinator, is produced by viola. Viola is an example of clistogamous flower, means the flower which never opened. Okay, next, which of the following is absent in gymnosperm? This should be all of this. Triploid endosperm. Gymnosperm will have haploid endosperm. Okay, free living gametophyte. What about this? In the case of gymnosperm, male gametophyte. Male gametophyte in case of gymnosperm means pollen grain. Okay, and pollen grain, the number of cell will be limited. It can be 3, it can be 4, or it can be 5. So male gametophyte, the pollen grain will be limited, and male gametophyte does not have independent existence. And female gametophyte in gymnosperm, female gametophyte in gymnosperm is again endosperm. So this endosperm and this pollen grain, which are male gametophyte and female gametophyte, they do not have independent existence in gymnosperm. They do not have independent existence. So here, free living gametophyte. So they don't have free living gametophyte. And in case of gymnosperm, again, the seed is naked because it does not have fruit. Fruit is absent in the case of gymnosperm. So that's why it does not have covered seed. Next, ex situ conservation includes. Ex situ conservation will be gene bank and tissue culture and wildlife safari park. This will be 2, means 1 and 2. 1 and 2, and this 2 will be in situ conservation in its own natural habitat. 1 and 2. Next question. Thymos type of inflorescence, main axis terminates in a flower. Main axis terminates in a flower. This is correct. And flower are borne in a basipetal order. This is correct. That means 1 and 3 is correct. That means answer should be 2. Suppose in the case of cymose inflorescence, this is the floral axis, the peduncle. The floral axis is going, and at the terminal end of the floral axis, it will develop a flower. Okay, so main axis terminates in a flower. Next, what happened when this peduncle, when they are about to, since it is only terminated by a flower in the between here, it cannot grow further. So that's why they can have branching from the side. And this floral axis means peduncle. Floral axis means peduncle. This floral axis, which is peduncle, it terminates with a flower here, which will be always a solitary flower. And a peduncle will have a branches here. So since this flower is developed earlier, the later on some other flower will develop, which will be of course younger to this. So this is terminating a solitary flower here. This is the first case. Peduncle means the floral axis. And flowers borne a basipetal order means what? The base one, the middle one is the most matured one, and the side one are younger. Okay, so this is called as a basipetal order. So 1 and 3 are correct. Next. Genetic codon is said to be degenerate. That means degeneracy means some amino acid are coded by more than one codon. This is called as a degeneracy. Since amino acid is 20, genetic codon is 64. Out of that, 3 are stop codon, but 61 will code for 20 amino acid. That means one amino acid can be coded by more than one triplet codon. That is called as a degeneracy. Consider the following matching pairs: chromosome and DNA doubling. This is correct. S phased. Action of the enzyme recombinase. This should be pectitin. This should be pectitin. That means this is wrong. Study of morphology of chromosome should be metaphase. This is correct. Pairing between homologous chromosomes should be synapsis. This is also correct. That means this is correct, this is correct, this is correct. But here the option is not matching. So actually A, C, D should be correct, but option is not there. That's why we'll take this to C and the D. C and D. This will be the answer. But chromosome and DNA doubling will occur during S phase. This is also correct. Okay. 145. The interaction between Mediterranean orchid ophidians and bees is an example of mutualism. In phenylketonuria, the affected individual lacks an enzyme that convert amino acid phenylalanine into tyrosine. And the enzyme related will be phenylalanine hydroxylase, which is an autosomal. The related gene will be an autosomal recessive gene. Which of the following is a connecting link between carbohydrate and proteins? This is given in the NCERT as pyruvic acid. Pyruvic acid. Connecting link between carbohydrate and protein means from the glucose it will form pyruvate. In the case of glycolysis, from pyruvate it will form acetyl coenzyme A. Then it will have Krebs cycle, and then it will have ETS, and then it will have oxidative phosphorylation. Okay, this is for glucose pathway. And if it is protein, then protein will break down to form amino acid, and amino acid will be converted to form pyruvate, and from pyruvate it will convert to form acetyl coenzyme A. This will be pyruvic acid. 147.2. 148. Cuscuta is an example of ectoparasitism. 3. 149. The formation of which of the following is an example of dedifferentiation? That will be interfascicular cambium. Interfascicular cambium is formed by dedifferentiation of parenchyma in the medullary raised. So this will be interfascicular cambium dedifferentiation. And this two, primary xylem and the primary phloem, this two will be primary permanent tissue. Primary permanent tissue. This will not have dedifferentiation. Question is asking dedifferentiation. Okay, next is 150, last one. The Punnett square is a graphical representation to calculate the probability of all possible genotype as well as the offsprings. This should be the answer. In the Punnett square, we are putting male gamete at one side and female gamete at the other side. And it is crossed and the offsprings are placed here. Okay, so that offsprings which is formed, the genotype of the offspring is represented by that square. So in that whole square, it is representing the gamete also, and at the same time the genotype of the offsprings also. Here what is meant matching is the genotype of the offsprings will be seen in the Punnett square. So this will be the answer. Okay. Now with this we end with the question discussion. Thank you.